Protista and then fungi, we are done for this semester, right? So you should be ahead of me because as soon as I finish fungi, the day after that we have a test, right?
Sure.
So really these 2 weeks you guys need to work hard. Read these chapters ahead. Be ready. I don't know why they did this, but we used to have a finals week. You had 1 week, just you didn't go to your class and just take your finals. But since they cut it to 16 weeks, we don't have that final week. All right. You already know a bunch of stuff about prokaryotes. We're going to expand on it a little bit. So we're going to look at bacteria and archaea a little bit. Learn some diversity. And if I were you, wherever I go, I would take one micro class because this is a huge part of biology. And there's a lot of good information. Usually these classes are fun. The labs are really fun. And gives you a different perspective. So whether you're a physiology major, whatever major you pick, take a biology, microbiology. All right? So we said bacteria and archaea, these are prokaryotic type of cells, okay, prokaryotes. And when we talk about prokaryotes, mainly means they don't have what?
Nucleus.
They don't have nucleus, they don't have any nuclear bound organelles, okay? That's what distinguishes a prokaryote from a eukaryote. And they have been around longer than any other organism on this planet. 3.8 billion years maybe, 3.5 billion years. And they were on their own for a long time on this planet Earth. Now, prokaryotes are very small, okay? Today you're going to observe, look at under our microscopes, you'll see the little dots, okay, that's the organism. Very small, but at the same time, very successful. How do we measure success in biology?
Reproductivity, survivability.
Number of offsprings, what else? Survivability, what does that mean?
How they can sustain in their environment.
How they can sustain in their environments. Anything else you can think of? Success.
How well they adapt.
What?
How well they adapt to their environment.
How well they adapt. How do you measure how well they adapt?
3 things we just discussed.
Basically, number of offspring who survives, right? That's adaptability, in a sense. Surviving, if you're measuring success, that's what you measure. The other thing we look at is how well they have dispersed throughout the planet Earth. So when you look at prokaryotes, you can find them from the most extreme environments, coldest, hottest, places that there is no oxygen, there is oxygen, places that is really acidic, that no other living thing can survive. These guys have been able to take over. So all of those put together makes them really interesting organisms and very well, in a sense, adapted to a variety of conditions. When we look at prokaryotes, most of them are what? Unicellular, made up of one cell. So there are some species that form colonies. I'll give you some examples. A bunch of cells that live together, we call those colonies. And even some of these cells may have become specialized in some way. Okay. As I said, they're very small, 0.5 to 5 micrometer. Okay. And when you compare those with the eukaryotes are much larger. Maybe I have a picture to show you, but I don't.
Are you serious?
Alright, that's good. When you look at prokaryotes, they come in a variety of forms and shapes. different name. I'm going to ask you guys to remember three very important ones, or major ones. This is what I'm looking for. If this is under microscope, this is a eukaryotic cell. That's the nucleus. This would be like your T cells. These are bacterial cells. Okay? That's how small they are. And this is probably 1,000 times larger. Okay? So compare a eukaryote to the prokaryote. But they do have physiology, they have a lot of chemistry, and they reproduce all of those good stuff that a living thing does. Alright? I already mentioned they like nuclei, they like membrane-bound organelles. They do have plasma membrane. In addition to plasma membrane, they have cell wall. Their cell wall is different than what? Plants. And it's different than fungus. So look at their cell wall, the structure, and learn a little bit about that. Okay? If you look at the cell wall of prokaryotes, there's two different types, if you will. Okay? And these types also are used for identification and grouping, but when you look at the top one, which we refer to them as gram-negative group, Gram was the person who created a special way of staining and then allowed you to be able to distinguish this easily, all right? So when you look at these two different parts of the structure of the eukaryote, the cell wall, obviously you see a big difference. Over there we have the peptidoglycan layer, and then this is the phospholipid layer. It's the membrane, the plasma membrane, plus the proteins and stuff. Okay, peptidoglycan, that's sugar plus peptide. What is a peptide?
Protein.
It's a bunch of amino acids, right? Stuck together, and then some sugars attached to it. When you look at a gram-positive type of the cell wall, what do we have? The peptidoglycan right here, okay? Then you have the plasma membrane just like that one, but on top of this we have another layer of what? Phospholipids and protein that looks like the membrane. You see the difference? Yeah. Alright? So these cell walls are obviously protective layer, and they are pretty tough actually to break. To just give you an example, if I want to lyse some human cells, some eukaryotic cells, and get the DNA out, all I need to do is dump it into what? Water. Some pure water, distilled water, right? The water goes in, blows up. You can't do that with these guys. You have to break the cell wall, and there are a variety of different ways. There are enzymes that we can use to break the wall. And where do we get these enzymes? We produce them actually. Your tears, these are protective ways to actually reduce the number of bacteria that can colonize a body. So your tears have that enzyme, okay? And other organisms. But what it does is it chews up, breaks down the cell wall, and then the bacteria flows up. Other ways, you can put them under pressure, okay? Pressurize it, and then you blow up the whole cell. One group of bacteria, one time, we were working with to break down, and we couldn't use pressure, so we had to use glass beads and then shake it. You throw them with a bunch of beads, you shake it really hard, you freeze it, you thaw it again, and you shake it really hard, and those beads will go and hit these peptidoglycans and then destroy it, opens it up. Alright? But eukaryotic cells don't have this. Much easier to actually break them apart, especially animal cells. Alright? Any questions about this? Now, we're not going to do gram sustaining this class. I'll talk about the class later. Not really designed for this kind of work. But this is used for distinguishing these organisms from each other, and it's useful for people who want to figure out what bacteria this is. This would be a very first step. One of the first things that you would do if you want to find out what kind of bacteria you're dealing with would be to gram stain it, because it puts you into two different routes, and then you have to do a bunch of other tests. By looking at the bacteria, you cannot tell what they are, right? They're tiny, very small, many of them shaped the same way, so you have to do tons of chemical analysis to pinpoint the particular strain or species of bacteria or eukaryote. All right? If you take many micro classes, especially the ones that are designed for nursing medical school, they give you an unknown, and then you have to figure out what this organism is. That's the fun part of that lab, problem solving. Okay? And this is one of those tests that you would do. Now, how do they look at the microscope after you do this gram stain? You use this couple of different mixture of stains. Gram positive ones, they come out purple because of the way the cell wall is set up, and gram negative ones will come out pink. Okay? Purple or pink, that tells you if it's a gram positive or negative. Over here, what kind do you have? Gram positive or negative? Both. You have both, right? So you have tons of them. You have a bunch of purple ones and some pinkish color that indicates both. All right, in terms of shape, come in a variety of forms and shapes, okay? We're going to look at maybe 2 or 3 of them. You have, now these pictures are really neat, but you should remember that this is electron microscopy. This is 100,000 times enlarged. Today you're going to look at some of these, but they're not going to look as pretty as what you're looking here. Okay? Cocci, these are carcass shaped. They are like a ball. Okay? Round, top picture. Those are carcass or cocci. Bacilli, slightly elongated. Second picture. Okay? And within that you have variations. And then spirochete or spiral would be another group. And again, there are some variations within this spiral type. Okay? So these are some of the very general shapes of prokaryotes, and as I said, there are more, but these are the major ones. Right, so remember them. I may show you pictures and ask you what it is, what shape is this, and so on. So these are the things that you want to know for practical. Now, under a spiral, you can have a spirochetes, which is a variation of the spiral form. All right? Another view. Okay. You already know that these prokaryotes, the bacteria or archaea, they do move around. Not all of them, but many of them are mobile. They do move from one space to another in an aqueous environment, as if they're swimming. So they have flagella, or flagellum for one, or many. And they use that to swim, to move around. There are some neat tests that you can do when you take micro class that helps you indicate if this organism has the capability of moving or not. Okay? You just put them in particular agar and fluid in it, and if they move, the color changes, and then that tells you that they're mobile. All right? They have these. The flagella is pretty complicated. This is one of the things that today the people, scientific creationists, or the special design people bring up, that this is so complicated that nature or natural selection could have created. And they do this all the time. With the things that we don't know exactly how it happened, or they bring it up, we don't know. But then we solve it, they find something else, say, well, this one we don't know. Right? There's a lot of things you know. But the eye was one of those things they used to bring it up, that eye is so complicated, the eye of the organism. And now we have a very good idea of how eye has evolved as we presented for us. Okay? So that one is not taken. So don't get fooled with these types of examples. But it isn't complicated. It's a series of proteins that have to be aligned in a perfect way for the tail to fit into the cell wall and be able to use for movement and motion. This is a good picture. Again, electron microscopy. This is the organism, and this is the tail, the flagella that it uses to move. All right. And your book has, let me see if I can play this. This is again under electron microscopy. This is salmonella, what type of bacteria you find in chicken and poultry. How they move. And they have a variety of different movement, motion for different group of bacteria. Or Archaea. All right. Are we having fun? Yeah. All right. Good. So the movement is called a taxi. That type of a movement. It's not really toward a particular direction, right? And as I said, it's pretty complicated. We need a series of proteins to set this up and organize it, and we don't know exactly how this particular structure has evolved. Other structures you find on prokaryote cells is fimbria, pili, those are different form of this, but what it is extension of that particular cell. So this is one cell right here, and now you have these hair-like structures that are extending from the cell, and they use that to attach themselves to different surface. So it's used for attachment. I may show you a picture like this and ask you what are these things, or I may ask you what is the purpose of these things. You need to know the structure, all right? For instance, in your mouth, you have a bunch of bacteria, and they use this to attach to your gum, your teeth, and that's why you need to brush, break these attachments. All right? Another structure, we didn't talk about this one, is the capsule. Now, under this type of microscope, please listen, because you guys are going to be looking at a slide. This is, you're looking at a bunch of bacterial cells, okay? These are the actual cells that are stained. The capsule is the white area around them, okay? That white area is the capsule, and the stain doesn't stick to it, and that's why it looks like that. Okay? This is a special type of staining. You stain it so that you can actually see if they have capsules or not. Yes? So if I ask you what is this structure, the clear structure around these cells, it's a capsule. So what is a capsule? Capsule, you don't see it in all the prokaryotes, but some of them have it. And in a sense, it's a layer that develops on top of their cell wall. Another layer, the polysaccharide mix. Could be protein, or it could be mixture. And what it is to hide all the antigens, to fool the immune system. All right, so it's a way of covering themselves so that when they invade an organism, their immune system doesn't see it as foreign. Okay, so it's a trick. not everyone does this, but some of them do have capsule. This is what you see with our type of microscope. This is what? Electron microscopy. Okay. Another view. Okay, so what is the shape of this? It's a rod shape. It's a bacilli, right? And the white area is the capsule. Another type of structure that some microorganisms make and form is called endospores. Okay, for short, sometimes they call it spores. Endospore. This is a way to become dormant. It's a very tough layer they put around themselves, those that can do it. A tough layer that keeps the organism and its genetic material inside, and they can become dormant for thousands of years. Days, months, thousands. And it's a good way of preserving yourself if there is not enough food. If the temperature is not right, if there's not water, conditions are bad, harsh, you form your endospores, you sit there till what? The conditions improve and get better. And they found some of these on mummies that are thousands of years old, and then they create the conditions for them. They actually started propagating and forming colonies. Okay? So we know that they could be preserved for a long time. The other thing that is important to mention, because it's such a tough thing, those that make this, it's very difficult to kill. All right? So there are some that grow in our food, a particular group that you could have it in your food, and usually when you boil or you cook your food, high temperature, you kill bacteria and no problem, kill the prokaryotes. But if they produce endospore, it's very difficult, right? You have to boil them for many hours to be able to destroy it. And then they could cause food poisoning. The other thing that these are used are for biological weapons, okay? So this is actually what was being sent, what was it, 6, 7 years ago, maybe longer?
Right after 9/11.
So more than 10 years ago. It goes fast. The older you get, the faster it goes. You know what, it was 3 years ago. No, it was 10 years ago. So you can refine these, and as the person who was sending these, it was a very refined powder style, put in an envelope, sent it to people. Several people died. Since then, we have developed some drugs that work pretty well, okay, against this. But this is one potential thing that can be used for biological weapons. As I mentioned last time, biological weapons have been around for a long time. People used it, giving blankets to Native Americans and so on, and killed a lot of them, just tricking them that this is a gift. And that's blankets that were used by people who had smallpox and things like that. Today, become more sophisticated, we can refine these and use them. We can even, through genetic engineering, make these much more dangerous and harmful. And it's easy to do. You don't need too many equipment, etc. You know, lab like this, a centrifuge, etc. You can do it. So it's much more, the potential for it is there. And they used to have, United States had a whole program on this, Russia had a program on this, many other countries had, they were highly, biological weapons. And then at some point they decided we should get rid of them, they're dangerous. So I don't know if they did, nobody is really monitoring any of them. So US may have their own pie, Russians may have their own, and other countries still have their own. The person who was sending these around,